Good evening, good morning, everybody. It is Saturday, the 17th of April, and you've tuned in once again to Cyberhuman Frontiers with us. We got a yet another view and another perspective of the advance of technology into the enhancement and manipulation of life. There's just so many parts to this puzzle. As I review and screen potential sources for information, I find it's overwhelming how much there is out there. And I'm watching for thank you, chat participants out there. I'm watching your comments, and just let me know that the sound is good, and I'll see that, and we'll continue on. Before we get too deep into it, I will just necessarily have to go through as quickly as I possibly can the community guidelines disclaimer, which is this. I'm going to read fast. The points of view and purpose of this video is not to induce fear or incite forms of violence, harassment, bullying in any way, but to share and exchange thoughts. And this is with other like-minded individuals who are curious about the subject. This is to encourage conversation and to grow awareness in the whole field. And you're going to find if you relate to something that we've discussed, then if you hear about something about it in the news, then it's going to ring a bell. And you're going to say, wow, I heard about that on Cyberhuman Frontiers. So that's kind of the whole purpose of it. So without expounding too much on the community guidelines disclaimer here, let me just get right to it. The opinions expressed in this video belong solely to the persons espousing them and are not a representation of the channel Cyberhuman Frontiers. None of our videos claim to give medical, legal, political, or financial advice. The content is created entirely for your entertainment, and we could call it technical entertainment if you wish. This video and our channel in general may from time to time contain certain copyrighted works not specifically authorized to be used by the copyright holder, but which we believe in good faith are protected by federal law and the fair use doctrine for one of the more reasons noted in the Copyright Act of 1976. So thank you for that. And here we go. So our general thrust here tonight, as it began to emerge to me, there is such a nexus of different technologies all converging on accomplishing some of the same things and perhaps helping each other to accomplish those things. What do I mean by this? All right, we've talked about so many things on this show. We've talked about 5G technology. We've talked about electromagnetics. We've talked about optogenetics, and we've done that recently, just in the last few episodes. And we keep coming back to this one central target, which is the code of life and the control of life, or the redesign of life forms, organisms. One of the central targets to this whole thing has been over and over again, the brain, the human brain, over and over and over again. That keeps coming up. Now, there are many ways to get into it and to conduct a therapeutic on it, or a modification or an enhancement. Some of them are more electromagnetic. Some of them are more mechanical, if you will, with nano-sized designed devices. All right. Other ways are by way of just going in and modifying the genetic code that controls what the cell is going to do. In other words, get that cell to build something on command by the designer of the code that took, let's say, commandeered some of the factory operations of a cell to make it do what it's going to do. And yep, this is live, by the way, and good to see you guys out there. I'm watching, and be aware that when and if I'm sharing a screen, I can't see the chat in order to respond to it, but I can still read it. So under all conditions, we can make you part of this exploration that we're on. So want to welcome our people in here. I have a couple of silent partners in the background, and that would be TMC and Chris Hunter, and Nemesis is in the background, and we'll see where we get to. I've kind of only got a kind of a loose direction for it, but here's the deal. We've learned a huge amount in the last decade or two about what nature has already built for us in our bodies and in our brains. We're moving forward at an increasing rate in the science of modifying, redesigning, and enhancing both the body and the brain. There's a lot of ways that it can go. Technology is powerful and it's advancing, and it can do good things. It can do bad things too. You can always keep that in your mind, that you would want to make it, you might want to make it your business to establish some sort of a monitoring of where the leaders in this technology are taking it and how they're using it. And I think that that's a job for all of us to do, because look at what's at stake. The deployment of whether it be genetic code or viral vectors or something more electronic into our world that will influence us. It could influence us in some very deep and profound ways, such as modifying the genes that we were born with in our own bodies. All right. All sorts of things like this. And it could even go further than that. If we're talking about alteration or editing or modification of genetic code, there are another kind of genetic cell that we have, which are called germ cells. Germ cells are our reproductive cells. So if modifications were to be performed on those, then that would create a kind of a modification to our progeny or our offspring. And so that becomes like a generational enhancement or a generational change that might stick with us for a really, really long time. So the stakes are really very high, and the potential for tremendous advancement in what we've come to know as the life form that we call human. It's huge potential, absolutely huge. And I could go into a little bit of other stuff, but it's kind of peripheral. So it's all about genes, the code, and the software. We run software on computers, but there's something almost exactly like that, but a lot more complex. That is the software that runs the little machines that are every one of the cells in our bodies. And again, one of the central areas of concentration has been the brain. That's the command and control center, and we've spent some episodes on that. And it's fantastic when we change those instructions, that genetic code. We can literally take command of a living organism, either at the cellular level or at the level of the entire body of it, to become whatever we want it to be. So we're going to show you some of the more promising methods, some of the ones that seem to be the most productive or the most accessible, or that at the forefront of the technology look like they're going to work in the near future for accomplishing these tasks: reprogramming, modification, enhancing. So we think you're going to find it fascinating. Anyway, guys, how are you? TMC, how are you today?
Indy, I'm doing good. That was a good opening monologue. I'm curious to see what the videos are that you have.
Yeah, well, we start off at Chris. How are you? Well, you'll see. Chris, how are you? I know that you're on mobile. How's your audio?
I'm doing mine wonderful. I heard
Okay.
you say nemesis. Is that like my arch nemesis?
No, that's just well, I don't know. You'd have to ask her. And she's in the background. She may want to weigh in on some of this at some point. And Chris, your audio is good on your device, so that's fine.
Cool. Thank you.
So let me start us off right away. All right, and I'll tell you where we're going a little bit ahead of time. You guys have heard of the field of molecular biology. Okay, so there's biology at the macroscopic scale, where you look at an animal. Then there's you get down into the smaller and smaller size. And when we're talking about molecular biology, that's kind of the core of where we're going with some of this stuff, which is the design and the study and design of of things at not only the atomic level, but the molecular level, and how that gives rise to the proteins, the structures, the neurological fiber, the muscle cells, all that stuff. That grows up from the science of molecular biology. And we'll feature a couple of people who are, who are studying and working in that field and what that means. But first, we're going to start off sort of at the macroscopic level. And this, I'll get this thing up here. And so this is we're going to start zooming in slowly into this field. All right, hang on just one second. I think we got it. I think I found it. You will find links to the feature presentations that we carry in the show notes and the links below. This one happens to be from UC Berkeley, and it's just an intro. So they're talking here about neural dust, a neural dust sensor that could be implanted into the body. All right, and now this is from the more mechanical, the outside engineered device. It's made out of little bits of metal and stuff like that, and it might have a transceiver in it that can be connected to outboard computer equipment. So this is pretty macroscopic, and this is the size of a little device. The state of the art for this in 2016 was about the size of what you see here. This is a very short video, but I kind of liked the zoom in that it does. So let's just play this. It kind of gets us into where we're going. I'm going to kind of narrate this. The music is pretty good, but it moves on very fast. So first of all, just to be clear, this was 5 years ago, and that device that you see there is smaller and more powerful now. It is at that point in time small enough to be attached to a nerve fiber, and that would then start to communicate, transmit and receive from that nerve and give a status reading outboard to external equipment. The potential for that is to help restore a paralyzed condition. Now I'm going to skip forward. They kind of ran through it real fast. The tiny devices could help quadriplegics. Yes, all that stuff. Yep, absolutely. Very small. It would record, this little device would record electrical activity from nerve cells in the brain. But if you followed some of our previous episodes, where we're actually at in the state of the art is having literally nano-sized designed little devices that are far smaller than a cell. So by scale, this is huge, huge. But even at this size, look what it can do. You could easily, with the scale here, you could have hundreds or thousands of these little devices implanted into various parts of your body for monitoring, assist, or control of parts of that body, including brain cells, including nerve cells in the brain. So, short video, they're going to show the waveform, they're going to show how they put them on there, and they're putting this thing under a microscope so you can look at it. I think they're going to show you an image. Okay, they're picking up the signal from this little device. It's actually communicating right now as they are connected to it. These patterns are really important because there's a kind of a signature pattern that comes from various parts of our body. A couple of the chief ones are the brain and the heart. And they have some characteristic types of electromagnetic signals that they radiate. And if those things can be recorded and characterized and can be corroborated with what we observe in the behavior of the individual or the organism, then you can start to build a database of, you know, what those waveforms mean relative to the state of the organism, particularly the human in some cases. So those are a couple of the chief radiating parts of the body, the heart and the brain, something to keep in the back of your mind. Now we have an example of how they were able to pick up neurological signals from the brain that were blocked somehow. And they were able to use those by picking them up, sending them out to a computing machine of some sort, and then having that computing machine control a prosthetic arm. I mean, this is a real person with a real prosthetic, and it's really working. And so this is all very macroscopic still. We're not down at that small scale yet. Newer and smaller neural dust. Well, where we've gotten to is way smaller than dust particles. It's nano-sized, okay? But yeah, so once again, 5 years ago, this is where they were. The future is here now, and it is advancing amazingly. I don't know if you caught that little bit, but using materials that could last a lifetime. So if you have implantable devices in you, once again, very small ones with current state of the art, these things will have a longevity that'll be as long as you. They can be in there with you, and they can be built in such a way that they're not rejected or captured by the immune system and expelled. They, you know, they just kind of coexist with you. And again, size of particles that we have now are small enough that you could have many, many, many of them inside of a cell. And they wouldn't be taking up so much space that they obstructed the functions of the cell. Yet, they could be utilized to, once again, to control that cell to do certain things, various things, whatever they might be. And we'll maybe get into that a little bit too. So look at this. These two things that you just saw, the heart and the digestive system, are part of what we call the autonomic nervous system. And it's autonomic because it kind of does what it does in the background. You know, you don't have to think about making your stomach digest or making your intestinal tract do its business. It's autonomic, just like your heart is. Your heart just beats. It just does what it does. You don't have to think about every single one of those heartbeats. They term that the autonomic system, yet being able to monitor it and being able to modify its function, you could perhaps restore better stomach function to somebody. You could restore better digestive action or a more regular heartbeat to somebody with, once again, tiny, tiny, tiny little devices. And that's if you didn't go in and reprogram the genetic code in the cells in that organ so that they started to work differently. That is at the molecular biology level, which is where we're going to end up tonight in the show. Berkeley, University of California. Thank you very much for that very short but very powerful little production. So there you go. Those are some of the things that we can do. We can restore a paralyzed function. We can help to smooth out or correct abnormal brain function in terms of epilepsy, things like that. All that type of stuff. But we're going to go deeper, and let me grab the next one here. It's keeping in mind that technology moves ahead so fast that it's like, wow, 5 years is a lifetime of development in materials science and research and development. It's so much can happen in that short time. So we have another little one here before we get into our other stuff. And I'm putting that up right now. And I happen to know that this one's a slightly different size. So I'm going to, oh, this one isn't. The other one's a different size. Okay. This is from, get it up here for you. This is from Cold Fusion. And thank you, Cold Fusion, for this very interesting little production. Here we go. We're not going to play all of this all at once. It's a little bit longer, but it'll give you an idea.
That nanobots could assist medical professionals in complex tasks after passing through tiny spaces in the human body. One of the biggest potential applications is for extremely targeted drug delivery to specific cells within the body. As we'll soon see, this has already been achieved.
And you will see the thumbnail that is associated with this show depicts one of those little nano-sized delivery capsules for bringing for targeting the delivery of a cancer-killing drug right to the target cells that it needs to get to. And that's so much better than the current state of the art with chemotherapy, for example, because it targets precisely and it doesn't damage the rest of the body in the process. That's a little bit of what he's talking about here. He goes on.
Scientists also believe that nanobots could be used to reduce plaque in veins, solve dietary issues, along with a whole slew of other medical uses. They'll be able to operate in parts of the body where the blood flow is slower, such as capillaries in the eye or fluids outside the circulatory system, such as ventricles of the brain or urinary tract. So all of you have probably heard of CRISPR.
I wonder if anyone ever really remembered. I got to review this. CRISPR, that is gene editing technology, and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Okay, CRISPR, gene editing technology. They've had it now for years.
Gene editing. Although not directly related, CRISPR is an offshoot of the nanobot idea. CRISPR is actually an exploitation of a natural molecular DNA function. Scientists use a virus's natural ability to cut and splice the DNA we want to edit at will.
And just to pause there, where we're really going is probably not as much reliance on something macroscopic as we used to. Because, in other words, delivering little bits of genetic code into cells so that we can insert pieces of perhaps an individual's genetic code was mutated somehow and it was missing something that it needed to have. Well, if we can figure out where that is, and they're feverishly working on mapping out every single aspect of the human genome, then it would be possible to know exactly where the missing piece was and put that piece in to bring about the synthesis of a protein that you need, for example, whatever it might be, an enzyme, a protein, or a structure that somehow an individual's body just, it was missing that piece of the programming, the code.
Talked about CRISPR DNA editing in the case of destroying cancer in a previous video. I'll leave it in the description below so you can open it in a new tab. We'll take a closer look at DNA nanobots in a second, but first, let's delve into where this idea came from in the first place. Information could be encoded.
We're going to skip ahead a little bit because you'll be able to watch this whole video yourself if you'd like to. So I'm just going to skip forward just a little bit. Want to get to scale here, which is right about here. Okay.
Technology conducted at the nanoscale, which is about 1 to 100 nanometers. Let's put that into a perspective. The ratio of the Earth to a marble is roughly the same ratio as a meter to a nanometer.
Okay, let that sink in. We're talking about something the size of a nanometer. So now if we equate the size of that cell to something that is nano-sized, let's say the Earth was a cell, and now we look at a nanobot or a nano device or a nano-sized object. Let's say it was designed to be inside of a virus as a delivery package. It would be really, 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 really small.
A nanometer is 1,000,000 times smaller than the length of an ant. A strand of DNA is 2.5 nanometers in diameter, making...
Okay, one strand of DNA, very small. But can you imagine that at this scale, this is a string of DNA it's not coiled up, it's not rolled up. imagine being able to reach this level and to go in there with that CRISPR technology and right where my mouse pointer is, find a little part of that DNA and identify that that's where a new piece of sequence needs to be put in so that you can bring back the function to, once again, to synthesize a particular protein that a person may need, whatever it might be, or any of a number of possibilities.
Making them the perfect candidate for a basis for nanorobots. There's a couple of types of nanobots in existence today, but right now we'll focus on those made from DNA and those that are man-made.
Okay, this is precisely where we're going in tonight's show, because there are two ways to do this. You can man-made devices or things that are made out utilizing the cell's own genetic coding factory machinery to build it. So this is exactly because in my view, that's where it's headed. Why would you go out and have to build all sorts of little machines and then get them into the body of a human or an organism when you could just put in some programming, some code, which uses the factories, every cell is a factory of sorts, uses the body's own factories to build what you want it to build. Then all you have to do is just bring in the information, the instruction. You don't have to actually, inject particles. So that's where it's really pretty well going. And it's quite elegant and it's extremely powerful. So anyway, here he goes.
But there's a couple of types of nanobots in existence today, but right now we'll focus on those made from DNA and those that are man-made. So let's take a look at the state of the art. Researchers at the University of San Diego developed nanorobots capable of cleaning toxins from...
Notice, it is cell-like nanorobots. Now that is not a cellular component. That is not something that is natural. It is a cell-like nanorobot. In other words, it's artificially designed. Okay.
Blood generated by harmful bacteria. These nanorobots are about 25 times smaller than the width of a human hair and are made from a combination of natural red blood cell and platelet membranes and artificial gold wire. They can be controlled by ultrasound, which eliminates...
Okay. Tiny, tiny, tiny little piece of gold wire in there, which then opens up that opportunity to control it with some of our electronic technology, electromagnetics, and TMC could expound on other ways that we can use. Once we've built something that's got any sort of an electronic receiver element in it, such as this tiny piece of gold wire, now we can do external operations with it. We can access it externally. That's different from reprogramming a cell to do what you want it to do by changing its genetic programming. This is artificial control. So there's two different avenues here. And they both work, and there's development in both areas, both fields, and they converge in the human body, or in the mouse body, if you will.
Eliminates the need for a battery during propulsion. While still experimental, early tests have proved successful, with nanobot-treated blood containing 3 times less toxins and bacteria than untreated blood. The scientists note that, quote, "This is a proof-of-concept platform for diverse therapeutic and biodetoxification applications," end quote.
By using this natural biomaterial membrane coating our micro-robot, we can impart new functionality, new capability, like removal of pathogen and toxin from the body.
Right. So in a sense, what they're talking... Oh, it paused on a blank screen. Sorry about that.
Researchers at the Arizona State University.
In a sense, what they're talking about here is building little machines that are going in and doing cleanup work in a body, but they are artificial machines. They are not life forms. They've been designed, but nevertheless, still quite functional.
And the National Center for Nanoscience and Technology of the Chinese Academy of Scientists injected nanobots made from a folded sheet of DNA into the bloodstream of mice. To perform their study, first, scientists induced aggressive tumor growth in the mice. Once the tumor was growing, the nanobots were deployed to come to the rescue. Each nanobot is made from a flat, rectangular DNA sheet 90 nanometers by 60 nanometers in size. A key blood-clotting enzyme called thrombin is attached to the surface. Thrombin can block tumor blood flow by clotting the blood vessels that feed tumor growth, essentially leading to the tumor's death. First, an average of 4 thrombin molecules were attached to the flat DNA scaffold.
These are some of the things that we've shown you how they we've shown you patents and some of these structures that are made once again at nano size. So he's going into some detail about how they're going to use thrombin to starve out the blood flow to cancer cells and do it in a targeted way. So I'm going to skip ahead a little bit. Any comments from any of you guys out there, by the way? Let's see what you got. How you doing, Robert? Hi, Sir Jeff. Good to see you. Hi, Pano. Hi, Mr. Dynamix. TMC, any thoughts percolating through your mind at this moment?
Not at all. This is very interesting. I think what you put together so far is very good.
So, yep, the therapeutics are literally right around the corner. If you see what we're talking about, this is from 2 years ago, and they've already had they've got their design, their proof of concept, and their test in a mouse. They've already got that far. So there's some hugely good news on the way for humans in terms of mitigating and reversing cancerous processes in our bodies. And you're seeing it right now. Here's what they did. There's the mouse. There's the injection. Here is the cancer right there growing, okay? And it has commandeered a blood supply. And that's kind of what cancer cells do. They have a way of accessing and grappling onto capillaries and commandeering some of that blood supply for themselves. So by this targeted method, they've been able to starve off the blood supply to the cancer, and it becomes what they call necrotic. And necrotic basically means dead, basically dead, a little piece of dead scar tissue that's just it's not biologically active, and it's not growing, and it's not overtaking the body anymore. So here we go.
To 45 days. Healthy tissue was left untouched, and I think this is pretty good for such an early trial.
So again, by whatever technology we use, targeting, TMC, targeting is such a big deal, whether it be by the convergence of electromagnetic energy or by the convergence of multiple optical beams delivered by laser, targeting, or whether it be by designing a molecule in such a way that it has the exact right chemical affinity for a certain type of cell. And if you wanted to target cancer cells, you design that thing, as these guys did, so that it zeroes right in on that area, that high blood flow area where the cancer cell has commandeered blood supply, and then cut that supply off. Or you could design a molecule which has an affinity for neurons. And those things, you get those into a body, and they're going to go to all of the neurons in a particular area, depending on where you introduce them, or maybe you'd introduce them into the whole body. So targeting is a big deal. And then being able to pinpoint particular parts to perform a particular modification to whatever was going on at that cell, or that neuron, or that nerve, or that group of muscle cells, if it were the heart, for example. So targeting, and in many ways that they're working on doing this.
They also tried their system with lung cancer. The tumor tissue showed shrinkage after 2 weeks of treatment. Professor Hao Yan, one of the lead researchers, weighs in with his thoughts. I think we're much closer to real, practical medical applications of this technology. Combinations of different rationally designed nanorobots carrying various agents may help to accomplish the ultimate goal of cancer research, the eradication of solid tumors.
Isn't this wonderful? This is good news. You know, we get into some scary stuff on Cyberhuman Frontiers too. But yeah, there's some really good news. The potential is fantastic.
End quote. Yan and his collaborators are now actively pursuing clinical partners to further develop this technology. A nanobot device is roughly the size of a red blood cell or smaller.
A nanobot device the size of a red blood cell or smaller. So that's going to be capable of flowing through the smallest of the capillaries anywhere in our bodies. All right? Okay, keeping that in mind. Scale is important because some of the stuff that we talk about is super wicked small. It's so small you could have hundreds of them inside a cell. So the scale he's talking about here is a little larger. And why is that? Because they've designed it's artificially designed. It's not something at the molecular biology level. It's big. It's cell sized.
This is too small to add elements like a motor, computer chip, or camera. So bioengineered bots made from DNA are a common platform for such medical tasks. But as you'll see,
this animation is absolutely fantastic. And you know what? This is probably more accurate. What you can see on your screen is a very, very close representation of what really goes on at that level in our bodies and natural processes operating in ways like this too. So
and his collaborators are now actively pursuing.
Playing it again because I just like seeing the animation so much, so I'm indulging myself here. Check this out. Look at that. Notice how that little thing is climbing along. It's walking its way along. What we're seeing here in this animation is something being towed. It's being pulled. We've got a little filament attached to a big object, and this end of the little filament is literally walking along. It's at the microscopic level. This is pulling something like a tow truck, and it's bringing it to a location where it's got to be brought. Guys, what an amazing world. Am I too excited? I mean, I should calm down. I'm just a nerd, you know? Look at that. TMC, is that cool or what? Delivery system, how to bring things from one place to another.
Yeah, that cell looks like it has the Rona.
Yeah, could be anything. But I mean, this is pretty slow, right? I mean, it's walking, it's tootling along, you know, probably at like 100 nanometers a second. So it's going really slow. But if you've designed these things properly and you get them into a body, and then you give it a few days or a week for these little, for this big blue thing to get jockeyed into position wherever it's supposed to go, then they'll eventually all get to where they were supposed to get to. I wouldn't think that it's far-fetched to consider that this type of a mechanism could be used to bring something to particular neurons in the brain, and that they could be influenced to go where you want them to go. In other words, particular centers in the brain for a reason. Oh, I muted the sound. Sorry. I muted it because I wanted to talk over it. We'll go again here.
Camera. So bioengineered bots made from DNA are a common platform for such medical tasks. But as you'll see next, nanobots made from man-made materials.
Indy, you there?
Oh, I muted myself now. Bioengineered nanobots made of DNA. That's one category. Now he's going to show you the other category. A couple different types.
Materials are possible and are currently being used in research right now. So here we come to our featured story, which was the inspiration for this video. Not long ago, I was contacted from someone from the Max Planck Institute in Stuttgart, Germany. The team behind this next story has done a world first, this time using nano-robot vehicles instead of DNA. These were the first nanobot robot vehicles steered in real dense tissue, a pig's eye in this case. Usually, these tiny vehicles navigate through artificial demonstrational biological liquids, not real tissue. The bots were made out of iron spirals that would swim under the presence of a magnet. The trick.
So pretty simple, right? iron spirals that will swim in the presence of a magnet. But if they're spirals and you get them to spin, then what they'll do is they'll spin, and as they rotate, they move in a direction that's associated with the axis of the spiral. So, okay, more.
And the breakthrough was a special double coating that allowed for the bots to swim freely without damaging the surrounding tissue. The hope is to one day incorporate a drug delivery system into these devices.
Is that something or what? TMC, isn't it amazing?
Yeah, it looks like artificial sperm.
I hadn't thought of that, but I suppose that there is some similarity there, yeah.
Devices.
So what is the vision for this nanopropeller? What do you hope it can do one day?
So the vision of these nanopropeller is that one day we will load drug to the nanopropeller, and they will carry these drugs and propel through the vitreous to the retina and release drug very locally in a time.
Now you can do it by injecting these local to the eye and deliver this stuff to the retina, but with proper steering, with really precise steering, these things could be made to migrate. I'm drawing them with the mouse pointer on his arm. They could easily be made under the proper magnetic control to be brought to anywhere, and probably to a very, very small, pinpointed, navigated location as well.
Targeted way. Yeah, so this is the first time that we demonstrate it is possible to use these nanorobots to really penetrate real biological tissues.
So what?
Nanorobots penetrating biological tissues with a payload. So, guys, does this ring any bells? Let me see what people are saying out in the chat. Does this ring any bells? Penetrating dense biological tissue and drilling their way through one way or the other, or being steered or energized to move and carry something to a specific location. So, yep, I'm just doing a quick chat review. We never disregard all of the participation out there. It's really, it's really kind of a joint exploration. So, yep, I'm reviewing and reading. So now he's talking about the future, and I think we can guess what. Excuse me. I think we can guess what some of the future might be.
What does the future hold? Well, there's a lot of activity going on in this space. Scientists at MIT have recently developed optically powered batteryless nanobots equipped with sensors able to record and monitor chemicals like enzymes and hormones. The information.
So, we've also discussed this. I think it was between 2 and 3 episodes ago, but we're talking about the optical side of this picture now. Okay.
It can then be downloaded and analyzed. The study has been hailed as a completely new field of robotics. Further to this, the California Institute of Technology are throwing their hat in the ring. They're developing DNA robot toolkits that are being created to allow medical developers to use them in building more capable DNA robots. The device is made from a single strand of DNA that can autonomously navigate around a surface.
So, with DNA robots, guys, DNA is the central code that controls everything that we are, grow into, become, or were designed by nature or our parents to be. So this is the hugeness of this. It's, wow. And there's another aspect of it that we haven't even touched on, which is to basically transplant pieces of code from other types of organisms and deliver those into a secondary organism. So, take something from, let me come up with an example. Well, there are some characteristics about sharks that for some reason they never, excuse me, they never seem to get cancer. And if we were to locate the specific genes or the specific characteristics of shark cells and identify certain proteins, and then transport or transfect those into human cells, then maybe we could start to pick up some of those cancer generating characteristics that shark cells have. There are literally thousands of examples of what can be done, and other ways of enhancing humans, almost in a way that's not present in any other life form on the planet as well. So let's see where he goes here next. These are brilliant people that work on this stuff. Absolutely brilliant. They're very creative. They have a vision, and they move incrementally ahead, one little step at a time, and they're really getting somewhere.
Medical developers to use them in building more capable DNA robots. The device is made from a single strand of DNA that can autonomously navigate around a surface, pick up certain molecules, and drop them off at predetermined locations. The California team used a DNA nanobot to successfully sort 6 scattered molecules into their pre-assigned places in 24 hours. Adding more robots would reduce the time needed to complete tasks. Lulu Quian, assistant professor of bioengineering, comments, quote, "It is my hope that other researchers could use these principles for exciting applications such as DNA robot for synthesizing therapeutic chemicals from its constituent parts in an artificial molecular factory, delivering a drug only when a specific signal is given in bloodstreams or cells, or sorting out molecular components in trash for recycling."
Okay, there's a lot in that little paragraph, okay? First of all, the idea of a DNA robot is that just blows me away to begin with. But using that DNA robot to synthesize a chemical, I know that she knows what she's talking about when she wrote that, but there's another way that this could happen as well. If you've got that DNA robot and you've been able to navigate it into a cell, kind of where I'm going is to get that to modify the functionality or the synthesis that the cell is undergoing so that the cell starts making a therapeutic chemical internally and then maybe releases it to the environment immediately around it for some purpose. There are lots of ways. There are so many ways that this can be done. And obviously they go with the ways that are the most accessible and easiest first. And then it becomes more complex, more precise, and more powerful. But that's her thought. Fantastic.
End quote. For some reason, it reminds me of open source development kits which other programmers can build off. This pattern of building off others is very commonplace wherever there's exponential scientific development. Okay, so let's recap. So we have DNA robots that can detoxify blood, other DNA nanorobots that can starve tumors, man-made nanorobots that can swim through dense tissue, tiny nanorobots that can sense its chemical environment within a body and relay that information to medical professionals, and a DNA nanorobot development kit. If we were to
Wow. Was that an earful?
compare the current situation to the historical progress of computers, this would be like the 1960s when mini-computers were reigning. They were hard to use, limited to research groups, not quite a revolution, but they showed great promise. Or this could be like the internet of the 1980s, where online communities existed, but it was before the web, and it was still when people were figuring out how this was all going to work. Although it's early in its life cycle, the progress of nanobots is moving rapidly. Maybe in 10 to 15 years, nanobots will become commonplace addition to the field of biotech, that is, if a meaningful way to reduce the future cost is found. So that just about wraps up our...
See what we got there? Did you see that little animation, that last one? So we have a nanobot right here, and it has been, let me roll it back a little bit if I can. Okay, it's coming in. Here it comes. It's coming in. Boop, 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 boop. All right. And watch what it does. It's got these little tentacles, and it knows where it's going, or it's being navigated or steered to a particular location, or it has a chemical or electrical attraction or affinity for a certain type of structure or cell in the body. It gets there. And wait for it. it grapples onto that structure, and it's going to now start doing something with it. It's going to modify it or take control of it, or maybe eradicate it too.
This has been Dagogo. You've been watching Cold Fusion. If you've just stumbled across this channel, feel free to subscribe. I'm sure you'll find some interesting stuff around here. Thanks for watching, and I'll see you again soon for the next video. Cheers.
Utterly fascinating. Guys, what do you think of this stuff?
Yeah, it looks great. The conceptual, I guess, artistic renderings, those are very good. I don't think they're actually going to look like that, but
no, it's a little bit messier in the body. There's in real application, there's a lot more fluid around of varying densities. It's a little cloudier. It's not as clear, and there's kind of actual biology is more like a soup of stuff, kind of like minestrone or something. You know, it's got some chunks, it's got some little ones, big ones, and it's got a cornucopia of different chemicals in it in an actual biological soup. But because of there's so much intelligence in it, because in that soup there are certain chemicals that tend to gravitate together and they start to form more complex molecules. That's kind of the way it is. It's much more mixed, and it looks chaotic from the outside, but it's actually got brilliant organization in it by the way that these molecules have been combined, and the way that they're all designed. They kind of work together. So anyway, I have more. Do you have, I've got another kind of an animation of stuff if you're interested. Or we could dive off into a few things that you might want to feature along this topic. I haven't gotten to the molecular biology part. I could do that now if you want.
Yeah, I think we should. This kind of stuff is important. I guess to kind of...
All right, I'll do it.
Yeah, this is... Okay,
so we've so far featured something more from the standpoint of, well, it's not even macroscopic. Some of it got pretty microscopic. Let me get into a little bit more about some of these delivery systems. And we're going to introduce a term, and the term is called a plasmid. What is a plasmid? And I'll give you a couple hints. It's going to be associated with virus particles. It's going to be associated with having something that you want to deliver to a location, but using an encapsulation called a plasmid to get it there. And here we go. Now, once again, giving thanks to the people who have, brilliant people who have made the stuff. This is from a channel called Add Gene, and it's fairly short, and this will give us an orientation to what a plasmid is in relation to movement of chemicals and molecules from place to place and getting them where you want to get them. And they're going to start by showing you how you make a plasmid. Didn't you like that intro music? That was cool.
At their most basic level, plasmids are small, circular pieces of DNA that replicate independently from the host chromosomal DNA.
Hold it right there. Okay. Small, circular pieces of DNA that replicate independently from the much larger pieces of DNA, which are housed in the genes, which are really a whole... I mean, this is one page versus the whole Encyclopedia Britannica, all right? This is just one page of code, but that's what they do. They are... So just imagine if there were plasmids out there that can get into your cells and they're self-replicating and they just keep making themselves, but you don't... I'm getting into the, the darker side of things here. Let's say you don't know what this plasmid can do or what it does, but it keeps making itself, and it's using the nutritional soup in your cells to keep fueling itself and keep making itself. Well, all right. So we're talking about designing these things. Okay, we go on.
Compared to the millions or billions of bases that make up the entire genome, plasmids typically contain a couple thousand at most. They're relatively small, stable, and easy to manipulate.
By the way, that's a true number. The general size of the genome, the human genome, is in the numbers of billions of base pairs. So that puts some scale on that. All right. Every one of your cells has basically a copy, a replication of all of the genes that it would take to make your whole body. I don't know if you knew that, but the whole code is contained more or less to the large extent in every one of the cells of our body. And it's in that number, a very high number. This is just a couple thousand, tiny in perspective.
So where do plasmids come from? In nature, they're found in microbes like bacteria. In the 1940s, scientists began noticing that there are heritable cytoplasmic factors that could be transferred between cells, but there was no consensus on what they were or what to call them. It wasn't until 1952 that Nobel laureate Joshua Lederberg coined the term plasmid, a combination of the words cytoplasm and id, Latin for it. In nature, plasmids often contain genes that provide a competitive advantage, giving its host bacterium an ability that it didn't have before.
Ah, okay. So even though it's independently replicating, it just resides in cells, or in this case, in a bacterium. However, at such time as whatever the code that's in this plasmid is, makes that bacterium more adapted to its environment and more survivable, then it can express whatever is in its little set of 2,000 base pairs. And of course, it could work the other way too. It could express itself and make that bacterium less competitive, in which case that would spell the demise of both the bacterium and the plasmid. And then you wouldn't get so many of them. And they'd kind of weed themselves out of the population.
These benefits include antibiotic resistance, the wherewithal to survive in harsh environments.
So you see the importance of plasmids. They really do make changes in what the characteristics of a cell are.
Ability to wage war to gain an environmental advantage. But plasmids aren't just useful to bacteria. Because they're incredibly easy to manipulate, they've also made themselves indispensable to life scientists and bioengineers.
Now, it's totally not out of the question to design. We've got within our capacities now the ability to design a couple thousand base pair plasmid. That's easy. With the state of technology now, that's easy to do. It may take a little while, but it's not hard. Okay, and we also have the technology to take this plasmid and to package it in a protein and lipid coat, which we would otherwise call something that seems a whole lot like a virus, and then get it into the organism. It's all very, very easy stuff to do. So this is a great way to import code. And if you know where we're going with this, if we can import code that is going to alter the functionality of, in our case, a cell, then we can start making that cell do different stuff.
Plasmids created in the lab are known as constructs or vectors. To understand what plasmids can do, let's break down its parts using a Plasmid Map. All plasmids contain an origin of replication.
Now, he's not going to get too technical about this, but just bear with it. Keep your attention on it. It's just a few major components of what a plasmid is. So I'll read them first, and then he'll say them. The selectable marker, the promoter, the restriction site, the origin of replication, which is the starting point, and the antibiotic resistance gene. Antibiotic resistance gene. Okay. So when this thing gets made or replicated, this is where it breaks, and then it starts getting made, duplicated again. Boop, 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 boop. And when it gets all made, then it gets put back together into a loop. Isn't life amazing what it does? It does this stuff all by itself. I'm going to roll this back just a couple seconds so he can give you his version of it, which I'm sure will be quite good too.
Plasmid Map. All plasmids contain an origin of replication, or ORI. It tells the plasmid where to begin replication. Plasmids often contain genes that are advantageous for survival. One of the most common, naturally occurring types of genes is antibiotic resistance. When used in the lab, antibiotic-resistant genes allow scientists to separate out cells that contain plasmids from those that don't. The wonderful thing about plasmids is that they can be easily engineered and can introduce foreign DNA into cells through electroporation or other methods. Many plasmids
Indy.
I muted myself again. Yeah, it's all good. It's a really handy, easy to design delivery system. Thank you for that. I got to pay attention to that. Okay. Comments so far on this TMC?
Yeah, this is really interesting. I've never heard of these before until right now, and they're
handy, dude.
Yeah.
They're handy. They're easy to make. And they're well within the capacities of huge mega-sized companies to build these and code them.
Yeah,
it's cool.
Very interesting.
Okay, so it's simple and it's a short video.
Are designed so scientists can insert genes they want to be expressed in organisms. One way to do this is through restriction sites. Restriction enzymes recognize these sites and cut out the present gene like molecular scissors. Then, a different gene can be inserted into the site. Often, these restriction sites are located in what's called a multiple cloning site, a short segment of DNA that contains several restriction sites. This adds flexibility to the cloning process.
Okay, so now this is an example of things that we don't have to know. We surely do not need to know what each one of these acronyms means because we only, what we want to try to bring to you is a general picture of what the technology can do. So that's fine. He's giving us nice detail.
How is the inserted gene expressed? A promoter site, which is upstream of the inserted gene on the plasmid, acts as a green light that allows gene transcription. RNA polymerase binds to the promoter, moving along the strand. As it moves along...
Okay, so this is the fabrication process. This is the manufacturing process taking place. See all these little things that look like nails? They are available biomolecules that are floating around in the soup. And because there is a logic, there is an attraction, an affinity, a purpose in this code, then these are drawn from the soup onto this other unit, onto this segment of code, and thereby manufactures something that has duplicated the code. That is exactly how it works in our cells. This is how we build and synthesize every molecule, every protein, every enzyme that we make. It happens this way. This is how it's done. It's kind of like an assembly line. And we've got trillions of these little assembly lines that are working right now all the time, manufacturing everything that keeps us alive. So what if you change what keeps you alive and you do something to enhance the capacity or take away a capacity? Okay, and so anyway, here we go.
Along the strand, it creates a new strand of mRNA expressing the gene. The plasmid clone...
So if you remember from previous episodes, what then happens with once you've built a piece of messenger RNA, that then has it's got another location that it's going to go to, and it's going to go to the quote unquote ribosomes. And at the ribosomes, that's where the final protein or the final molecule is manufactured by way of the code that was brought from this thing to the mRNA to the ribosome. That's how it works.
The cloning process is very versatile, so there's a whole array of genes that scientists can introduce into the cell. For example, if you wanted to track a specific species of bacteria in a population, you could insert a GFP-expressing gene into a plasmid and transform it into your bacterium. Your cells will fluoresce, making them easier to find. Or if you're trying to study the effect of a specific protein on a phenotype, you could insert the gene of interest in.
By the way, just background information, the genotype is the actual code. The phenotype is what you see in the organism. That's the manifestation of what the code is. So terminology, you don't need to know all of it that these guys know, but that's the difference between genotype and phenotype.
To the plasmid. Protein find. Or if you're trying to study the effect of a specific protein on a phenotype, you could insert the gene of interest into the plasmid, move this into a cell, and look for changes in the cell. We hope you enjoyed this video in our Plasmids 101 series. To catch our new...
All right, so this is from AdGene. You will find credits and links to the fine people, brilliant people who do this research. Time for a little conversation here. TMC, what are you thinking? Look at the capability of this, and I got another one it's another short one that's on this subject as well. But just want to stop and see what your thoughts are. Just checking in with you.
It's a great mechanism for gene editing. I don't know enough about it to, I guess, understand the nuances between using this method versus
CRISPR.
Yeah, or some other viral delivery or whatever. I mean, this looks like this is perfect synthetic biology, build a DNA loop, and then plug it in with a couple other things, and then call it Pfizer or Moderna, or you know what I mean?
Yep. I guess one other thing that I wanted to, well, two thoughts. One is that CRISPR is the big guns. That's coming in with the big system. That's really powerful. And in terms of power, what we're looking at here is just a simple loop of a couple of thousand base pairs. A base pair is an individual unit of what would make up at a much larger scale a whole gene, a whole piece of genetic code. But this is only a couple of thousand of them. It's not many. And in terms of scale, if you visualize this one single simple loop, instead of this, if we want to look at the complexity of human genes, instead of a loop, think of it as a long coil. And then think of that coil almost like a braided rope, where you can look with a magnifying glass at the braided rope and see an individual strand, but then you see bundles of those strands that are subgroups, and then take that rope and coil it up around, or take a string or whatever it is, and coil it up around something else. And then once you've coiled it around another, say some sort of a flexible rod, take the flexible rod and coil that up too. So you've got coils of coils of coils. And that's the way that our human genes are when they're in their compact form, and when they're not replicating, when they're not duplicating or reproducing. That's the way they're stored. It's coils upon coils upon coils. And that's how you can get literally billions of base pairs into a very, very small space. That's just I don't know if that explanation brought any sort of an image to life in anybody's mind, but that's how they're stored in our cells.
For example, if you want...
So anyway, there we go. That was that one. And I have one more, which is students at a molecular biology lab. Students learning how to do this. And this is, I believe that this one is narrated by a student. So what comes to mind that we might want to, that from your huge bank of information that comes to mind for you that you might want to talk about, or thoughts or comments about some of the applications of this? It's such a convenient method for importing pieces of genetic code into cells.
Yeah, most of what I found looking at this kind of technology in the YouTube space really focuses and centers around targeted gene therapy, things like cancer treatment, and rare genetic diseases, things like that. But when you look at what this stuff can do, instead of looking at it specifically for what you're being shown, or this targets liver cancer or colon cancer or whatever, look at it as a platform. Look at it as a set of tools that becomes modular, that can then be used to selectively apply therapy anywhere in the body.
And you could do it additively too. For example, you could bring in, you could basically infect cells with a plasmid, all right, that's got a piece of the code that you want. And then you sit back and watch, and you look at what the cell is doing, right? And then you import another piece of code, which you've designed these two plasmids so that they're going to have some sort of complementary function, right? Or they're going to work together in what would start to become an increasingly sophisticated modification of the function of a particular type of cell in a body. And then, so you get the second one in there. And now you've got modification number 2, right? And you've started to see the behavior in the cells that you want. You've got it, it's at step 1 of a 5-step process. Now you just keep doing this, and you can keep adding and adding and adding information in there. And you've got it set so that they're all going to work together, right? You don't have to do it all in one step. Make any sense?
Oh yeah, I have a feeling that that's what the whole vaccine program is about, is exactly what we're more or less kind of dancing around right here. This idea that you can introduce a piece at a time and potentially have those individual pieces kind of start, recognizing each other and doing some additional changes, manipulations, functions in the body weeks or months or maybe even years later.
Years, could be years down the road. It could be just getting it parked in there, getting the code parked in there so that it really doesn't express until there is some sort of an environmental reason for it to have to express. But I'd like to take just a moment, if I could, just to indicate that this piece of content is for entertainment purposes only. And of course, we wouldn't want to say that word, because just saying that word might get our whole video banned. So, but we will also put the link up to our Rumble upload site here. We're going to be getting this stuff up there so that you can view it there. Should for any reason a video not be found here on this platform anymore. So she's going through, and she starts with Post-it notes showing how she's building this thing. This is also very short. You might find this interesting as well. We could run this unless you want to comment some more. But yeah, I agree with you. the ability to bring in new characteristics, new genotypes, in other words, new code that's going to, at some point in the future, express and have and result in a different character of a cell is it's super powerful. They're at the point of being able to literally design life forms, and we've touched on that before. It's amazing. I think I'll go ahead and play this video once again. This is from AdGene, and this I have got one here. This is from a student, and she had a project to do, which was to build a plasmid, and so she's going to build a plasmid.
The AAV vector has been developed for gene delivery both in vitro and in vivo. This video will cover how to identify an AAV plasmid and understand some of its important elements. In order to produce the AAV vector, 3 plasmids are required: the packaging plasmid, the transfer plasmid, and the helper plasmid. This video covers the transfer.
So I'm going to learn something. I don't know what the helper plasmid is, but I'll bet she's going to tell us.
Plasmid, which encodes the AAV vector genome. The AAV plasmid I'll review in this video is a basic AAV control plasmid encoding GFP. First question: how do I even know this is an AAV plasmid? By the ITRs, or inverted terminal repeats. AAV plasmids can be identified from other.
Okay, she's smart and she talks fast. She's firing off these acronyms here at us, and we got to pay attention to this one. Fortunately, it's only 4 minutes long, so we won't get too buried.
Non-AAV plasmids because they have 2 ITRs. The ITRs in the region.
Okay, ITR, inverted terminal repeat. Okay, all right.
Between are what gets packaged in an AAV molecule. Everything outside of this region is your plasmid backbone and does not make it into the AAV vector. This region within the ITRs will contain several elements. The most important is usually your transgene. This is the gene you're studying and want to get expressed, such as the
Okay, so there you go. That's what they're trying to get to, where they want to get it to, the transgene. All right, and it's not very big. These are small stuff. It's not a billion base pairs. It's only, 1,000, couple thousand.
Fluorescent protein GFP. In order for your gene of interest to be expressed in an organism, it'll also need to be downstream to a promoter sequence. On plasmid maps, promoters can be found upstream of the gene it controls. Promoters are specific to organisms and sometimes even tissue types. Using a fish synapsin promoter, for example, will result in expression specific to neurons in the fish.
Ah, okay. So it's going to be important to have the proper not the helper, but the promoter for whatever your type of cell that you're trying to get this into. Otherwise, it would most likely be rejected, and it would be recognized as a foreign body, and it would not be incorporated as you would want it to be incorporated. Okay. I'm sort of following along with her.
Using the same promoter in a mouse, however, will result in no expression. Promoters can also drive different levels of expression. Strong promoters like CAG will drive very high and non-specific expression levels. All AAV transfer plasmids will have ITRs, and most of them have promoters. Now I'm going to talk about some of the elements that aren't required for AAV transfer plasmids, but that you'll often see in them. I'll also describe what these elements do and how they might affect the activity of the AAV vector.
So TMC, how does this all relate to the general name of our whole operation here, Cyberhuman Frontiers? Well, computer science, using computers to quantify and to characterize huge groups of human base pairs, okay, billions, and then being able to go through that catalog, that index of base pairs, and zeroing in on the one that you want to do a change to or a modification to. And then having people such as this lady who are designing a particular delivery segment, a particular delivery package to bring that code to what has previously been cataloged by those other individuals who they know what every part of that human gene is responsible for doing. So it's a lot of numbers. It's a huge, it's like a phone book full of phone numbers. That's what genes are like. It's a whole phone book, 1,000 pages of phone numbers. And what we're talking about is at this level, we're talking about picking a particular page and a particular name and a particular phone number and saying, all right, I got this name associated with that number, and I'm going to change these numbers on this phone number, on this page, in this big phone book, and that's going to change the name, or that's going to change something about it. That's the scale. But we can handle this with powerful computers and good laboratories so that we can get in, and we're not really there yet, but we're getting to the point that we can get into any part of that human gene code, and with, once again, appropriating the time and the resources to get to the specific part that we want to get to, and then alter it, edit it, reprogram it somehow. So it's I could go on, but I'm going to let her go on.
In order to control gene activation, transfer plasmids will sometimes have lock sites. These are directional 34 base pair sequences that can be found flanking the transgene. The transgene is usually inverted to prevent expression.
She's so technical. Wow, she really understands this stuff. She's really smart. I have a lot of respect for that. She's learned, she's studied, and she's working in a lab, and she knows what all of the pieces of this are. And interesting thought, looking at life, it's all kind of modular once you get down to the small level, right? And it's really just which modules you decided to connect into the larger network, and what you're trying to make it do.
Lock sites are recognized by the recombinase Cre. Transfer plasmids will usually have two pairs of lock sites. This dual lock system is called flex or DIO. Cre will first flip the sequence between the first pair and then excise the sequence between the second pair. The result is a permanently flipped gene. This allows researchers to control the spatial and temporal activation of genes by controlling the availability of Cre. While lock sites are used,
spatial and temporal activation of genes by using the acronym that she just referred to. So look at the power. Look at this. And this type of thing could be used to exploit, to create a change in any type of a cell. Once again, it could be a connective tissue cell, a nerve cell, a muscle cell, or groups of them.
Useful for turning genes on and off, other elements can contribute to how well the gene is expressed. These are typically used to enhance the expression of a gene and are called Cis Regulatory Elements. I'll describe the WPRE and the poly...
Oh my God, she's so good on the terminology. She's blowing me away with the terminology because she's got it all at her fingertips.
A sequence, which are two examples of cis regulatory sequences commonly found in AAV transfer plasmids. Both of these elements usually appear at the end of the AAV genome, just upstream of the final ITR. The WPRE element helps the transcribed RNA form a tertiary structure and aids in nuclear export.
Okay, so that's kind of significant, right? Aids in nuclear transport, okay. And converts, I think she said, to change it from a tertiary structure, which would be a coiling. She's referring to the coiling up of that molecule. I'm going to play that again.
And aids in nuclear export. The poly-A sequence creates a tail of adenosine nucleotides on the mRNA. This also aids in nuclear export. Once in the cytoplasm, the poly-A tail stabilizes RNA by protecting it from degradation. The poly-A tail also aids in RNA translation. Both the poly-A and the WPRE sequences help increase the expression of your transgene. This video has shown you how to identify some of the key elements in an AAV transfer plasmid. Remember that there are many possible element combinations which will depend on the purpose of your AAV. When packaged into an AAV and delivered into a host, each element will contribute to the successful and specific expression of your gene of interest.
And there you have it, pretty much. All right, time for us to take over. Are you with me?
Yeah, I'm here with you, man.
All right, that's all I got. But it's just amazing. The takeaways from this are that there are people who are learning and working with this stuff, and they're coming ever and ever closer to the ability to modify life in so many different ways. And they're right down to the nuts and bolts of how you do it.
Yeah, I'm looking real quick for something that.
So while you're looking, I can go off on a little bit more commentary here, which was the title of this episode, which is Nature versus Science. All right, and we because our previous episodes were highlighting those exploits within the human brain and brain neurons. I kind of talked about that, but we have a tremendous opportunity to reverse engineer nature and look at what it, if you just leave it alone, what it does, and then to take pieces from one type of organism which have a characteristic. And one of the little video clips that we showed mentioned creating a kind of photoluminescence by bringing in a gene from a photoluminescent organism into one which was previously not photoluminescent, and then add that function into the cells in the new organ, in the modified organism. Well, that's just one example. That's photoluminescence. What if it was the production of some other type of protein that does something else? Or what if it's whole sections of code that are from a similar type organism but different? I'm trying to think of an example here. I don't know, one type of furry crawling creature versus another one, and combine certain characteristics, either in very, very tiny groups for the purpose of maybe helping something at a small scale, or something major, whereby let's say I put my science fiction cap on there. Let's say I take a mouse and I manage to combine together some of the attributes of some sort of an amphibian or a fish, and I can make a mouse that has both lungs and gills. So it can breathe underwater continually, or it can breathe out in the air continually, because it's now got the code combined from those two types of organisms. That's not impossible. Not impossible. So we've got groups of people who are working very, very diligently on defining and categorizing, indexing every single little part of the human genome. And that is the roadmap. That's the phone book. So you know how easy it really is to use a phone book and to look up a number. All you need is the name of the person that you're trying to find. And out of that whole 1,000 pages, because of the index, you're going to get right to the name that you want and get that number specifically and accurately. That's what they're doing with the human genome now. And they've done a lot of it. But in case you're interested, what they have initially done is to focus most of their attention on the parts of the human genome that are essential for human characteristics. And that's shockingly, only about 2 to 5% of all of the genetic code that's included in human chromosomes. And the other 95% has been, it's gotten a little bit less attention up until recent times. Why? Because of the sheer size of the task of categorizing and indexing it all. And because, we have far more powerful computers now than we did just, some few years ago. So now with this added capability, it's well within the capability of us now to be able to basically index the whole thing. And then it gets into the next aspect, which is, all right, little subtle differences between one race of humans versus another one. And that's some of the subtleties that we're starting to look at. There are organizations which go about tracing your ancestry, what your genetic heritage is. And it's all part of that same picture. And they're categorizing and cataloging and indexing all of that stuff. We're going to reach a point at which we have done that same type of categorization with many, many different types of creatures, and including humans, and then be able to mix and match genes from one to another. And this business that we've kind of featured here with the plasmids, this is one way of getting code from one organism into another. So, yeah. So, did you find what you were looking for, TMC? I know you're there.
Yeah, I found a couple articles that kind of loosely tied in. I sent them to you in.
I see something here.
Pentagon wants to use your DNA.
Where did you actually send it? Did you send? You must have sent it in Zoom, right?
Yeah, to Indigo Dot. I think it was your host account, not your co-host account.
Yeah. If you send it to the co-host, then I can put it up on that machine for display. Send that to the co-host. I'll get it right up.
Yeah, let me see if I can.
Yep. So, in so many ways, humanity is on the verge of massive new developments, and it's in flight. All right, it's happening now. It's well underway. All right, that's opening, and let's see here. Okay, I got it. Put this up. Oh, I see a mosquito. Hey, what's that mosquito going to do? Let me get this up on screen here. All right, and I'll scroll through this, and I'll just follow your lead on this one.
I'm trying to get this other one up here too.
All right, so we have here something. This is an article from The Guardian, a journalistic publication. And the title of this little article is U.S. military agency invests $100 million in genetic extinction technologies. Let me enlarge this a little bit more. Okay. Technology could be used to wipe out malaria-carrying mosquitoes and other pests. But UN experts say fears over possible military uses and unintended consequences strengthen the case for a ban. All right. And I'm going to have you take over on this. I got it up there.
Yeah, sure. So the U.S. military agency is investing $100 million in genetic extinction technologies that could wipe out malarial mosquitoes, invasive rodents, or other species. Emails released under the Freedom of Information rules show. Documents suggest that the U.S. Secretive Defense Advanced Research Projects Agency, DARPA, has become the world's largest funder of gene drive research and will raise tensions ahead of a UN expert committee meeting in Montreal beginning on Tuesday. I don't remember when this was dated. This was a few years ago. The UN Convention on Biological Diversity is debating whether to impose a moratorium on gene research next year, and several southern countries fear possible military application. UN diplomats confirm that the new email release would worsen the bad name of gene drives in some circles. Many countries will have concerns when this technology comes from DARPA, a U.S. military science agency. One said, the use of genetic extinction technologies and bioweapons is the stuff of nightmares. But known research is focused entirely on pest control and eradication. Cutting-edge gene editing tools such as CRISPR-Cas9 work by using a synthetic ribonucleic acid to cut into DNA strands and then insert, alter, or remove targeted traits. These might, for example, distort the sex ratio of mosquitoes to effectively wipe out malarial populations. I'm going to say that again. For the example they chose to use in this article is distort the sex ratio of mosquitoes from male to female, the ratio. Throw that out of whack.
Yep.
Deciding gender.
TMC, speak a little louder on that. I got a reading from someone. You were a little low on the decibel level there. I hear you.
Yeah. Okay.
Good.
Yeah. All right. So some UN experts, though, worry about unintended consequences. One told The Guardian, you may be able to remove viruses or an entire mosquito population, but that may have downstream ecological effects on species that depend on them.
Exactly.
My main worry, he added, is that we do something irreversible to the environment, despite our good intentions, before we fully appreciate the way this technology will work. Jim Thomas, co-director for the ETC group, which obtained the email, said the U.S. military influence they revealed would strengthen the case for a ban. The dual-use nature of altering and eradicating entire populations is as much a threat to peace and food security as it is a threat to ecosystems, he said. Militarization of gene drive funding may even contravene the NMOD Convention against hostile uses of environmental modification technologies. Unbelievable. Todd Hukin, who has worked with the GBIRD program, which received $6.4 million from DARPA, said that the U.S. military's centrality to gene tech funding meant that researchers who depend on grants for their research may reorient their projects to fit the narrow aims of these military agencies. Between 2008 and 2014, the U.S. government spent about $820 million on synthetic biology. Since 2012, most of this has come from DARPA and other military agencies, Kuiken says. In an email reporting a U.S. military-organized conference in June, U.S. government biologists noted that DARPA's biotechnology program manager, Renee Wesrigan, had said the Safe Genes projects account for $65 million, but then mentioned with all other support in the room, it was $100 million. A DARPA spokesman said that the figure was a liberal notional estimate that included researchers at the meeting funded by DARPA under related efforts. DARPA is not and should not be the only funder of gene editing research, but it is critical for the Department of Defense to defend its personnel and preserve military readiness.
Gee whiz. Okay, I understand the purpose of being able to protect yourself, and if we're not moving ahead with technology such as this, other people will, and who knows what they might deploy that eradicates one thing or another. It's I don't even know how you begin to get this back into the box and put the lid back on it, to be honest with you.
You don't?
I don't see a way.
Yeah, I don't
So all you can do is try to keep up with it.
Yeah, basically it says here that the cost of doing gene editing now is falling dramatically. And that's the concern that they have, is that hostile or rogue actors, it's well within a reasonable cost structure to start experimenting and researching.
Do you remember,
genetics.
I think it was about 8 months ago now, on a previous episode, we had, I think it was a TEDx talk, and there were a bunch of gene researchers up there, and one of them was a young guy, and he said, I want to make the ability for gene editing to be available for everybody at low cost. I want you to be able to do recombinant DNA research on E. coli at your kitchen table. You remember that? That was a long time ago. I'll bet you do remember it.
Yeah, I do. It was George Church and two other individuals that were, one of them was an MIT writer. He wrote for MIT's paper. And then it was the guy you're talking about. He worked for NASA for a number of years and then branched out into synthetic biology.
If you guys get a chance, apart from looking at we'll have more and more up on our Rumble site for archives. But if you get a chance to go to humaninternet.org, a lot of interesting stuff there, which is all relating one way or the other to this, and plus more on the computer side of things too. But nature has been building and balancing itself. I mean, this is like, I feel like I'm just saying something that's obvious that everybody knows, but nature's been building, rebuilding, and remaking itself for millennia. And if we come in there and we decide it's going to be good to get rid of the malaria mosquito, all right, there's something else in that whole network and interdependence of all of the creatures in the biological space that's going to be affected. And unless we really know what those effects are going to be, then we don't know what the outcome is going to be. And it's really easy to pick on a malaria-carrying mosquito, right? I mean, first of all, who even wants to get bit by a mosquito anyway? Let alone if it's carrying malaria. But it's not nature is our friend, not our enemy. And to tinker with it is probably sooner or later to inadvertently take away something that was helping us, and we don't know, because there's an interrelationship between many different species doing what they do in their interaction that could wind up having an effect on us, and a negative effect on us, I guess. So we should read the rest of this thing too. I think.
Yeah, Gene Drive is an interesting. There's a lot of stuff out there on Gene Drive. It's an interesting technology. But really,
and gene drive, to my understanding of it, is that is the push for certain genes in the living space to be promoted and multiplied and propagated, and what drives certain genes to be replicated and to be reproduced in descendant species, in descendant organisms, offspring. Is that what it is?
Kinda. A gene drive is more like a thumb drive with software on it that's going to be used to change information on a computer. So you're the computer, your genetics or a cell is the computer, the factory, everything. And then the gene drive is simply software that is inserted into the cell or into the host that goes in and changes the functionality of the factory, goes in and changes the functionality of the hardware.
And various ways of doing that. At the big scale with the big guns, CRISPR, and at the small scale with only 2,000 base pairs at a time, the plasmid method,
or combination of those things, because the CRISPR is simply just a cut and a paste tool. You start putting that technology together with something like a plasmid. And now you can go in and start selectively editing genetic sequences at particular locations.
Yep.
Where you're not just cutting cutting something open and removing it or disabling it, where you're actually going in there and potentially doing a reconstruction on it.
Modification, yep.
Or a remodel, yeah.
Yep. Could either be a slight one, or it could either be a very subtle, or it could be a very aggressive remodel.
Yeah.
And I'm thinking that the way that they're going to engage with this and implement this is going to be piecemeal. It's going to be a little bit at a time. You know, no drastic changes all at once. You know, when we're talking about that, when they consider the idea of eradicating a whole species of mosquito, that would be a drastic thing. But I think most of this is going to take place in very, very subtle ways. And that if it does become implemented in us, then we're going to find that there are subtle changes that take place that we might not notice, and they could be over a period of a number of years. And we wake up someday and we say, wow, I'm different now, but why? And so are we going to know everything that's being done? I'm not sure, especially if someone is, a kind of an independent creator of plasmid delivered gene code. How are you going to know who even did it? It's not documented. It wasn't done in a research institution or funded by DARPA and is, patented. It's just some little crew of people that built something, and they got it out there. Is it easy to deliver? Extremely easy to deliver. Extremely easy to deliver. Viruses are very handy delivery tools. Mosquitoes could be another one, I suppose, at a larger scale. Using mosquitoes to deliver viruses or just airborne. So when we've got a whole lot of new modified genetic code that's going to be floating around out there in the environment or flying around out there, we kind of don't have a clue what's going to happen. So let's finish this up. Gene drive research pioneered by an Imperial College London professor, Andrea Cristiani, who was confirmed has been hired by DARPA on a $2.5 million contract to identify and disable such gene drives. Fears that the research could be channeled toward bioweapons were all fantasy. I don't think so. I don't think so at all. And then he goes on to quote, "There's no way this technology could be used for any military purpose." I disagree. Don't you?
Yeah. I completely disagree.
How could it not be?
Exactly.
Well, exactly. So so he's he's trying to hold to a position, right, Chris, so as not to alarm anybody, and so as to perhaps continue to justify that nothing much to worry about. This is just normal research and development.
That's his attempt, but that's definitely not what we would believe.
No.
Possibilities are limitless.
Yep. Can you imagine, Chris? I mean, it's not even hard. We say this during this show frequently, but science fiction comes true. Can you easily come up with a few ideas of what you think could be a future that we'd be looking at? And what? Let's say those who have a particular type of genetic code modification to themselves versus others, and how that could drastically change nature has a balance of power. So drastic change to nature's balance of power, maybe?
I totally think so. I mentioned this before to you. I think that we have a deficit of memory and storage of our memory, and that to say genetically, I see animals that know how to do things. It's like I said before, I got baby chicks, for my kids on Easter and stuff, and they come in a box a day old, and they know how to do everything they need to do to be a chicken.
Yes.
Laying an egg, where to sleep, what to eat, what not to eat, cleaning, like they'll take a little bath in the dirt,
Yep.
flop around. But nobody tells them to do that because they were hatched. But humans don't really have that. And that's one of those things that I wonder what's turned off on us. I think that some people who are exceptionally smart or able to understand things, they're able to access memories, old memories, ancient memories that's stored in your DNA from previous, your lineage, like Leonardo da Vinci or Albert Einstein or whatever. They're able to perceive things that we really don't have access to or don't understand or are capable of doing, but we just don't have access to it. And I think that that's something that we'll hopefully be able to do is put memory into that and access it.
I want to offer something that's peripherally associated with that in my reading. I was doing a lot of reading today. So you making reference to this large bank of genetic code. It's a huge it's a wealth of information about life, and it resides in the animals and the plants of this earth. Well, it appears that there is more diversity in the bank of genetic code that's in other animals than there is in us. And I'll tell you why. Apparently, there was an extinction level event some 75 to 90,000 years ago. And if you ascribe to the idea that humans have been around for, 100,000 years or 500,000 years and not just for 5 or 10,000 years, I'm not trying to get into that. But if you ascribe to that and you go along with the idea that it seems that there was a human extinction level event that occurred about that long ago, well, there were only a very, very small number of human survivors, and that reduced the size of the library of information that we have versus many other animals. So it's not hard to find examples of that, manifestations of that as we see the phenotype or the expressed characteristics of other animals. They've got wings, they've got exoskeletons, they have other enzymes, some of them can glow in the dark, some of them can make silk out of a secretion, they've got fur, hair, all kinds of things that they have. So when we were talking about that library of DNA information, and with current technology, the ability to index more of it, when we do that with the rest of the plant and animal kingdom, and then we combine that with the ability to cross-transport various pieces of genetic code from one species to another, there is a gigantic encyclopedia of all sorts of code that can be used to change to change one organism and to import characteristics from one to another. And we, even though we're very advanced, we have a more limited encyclopedia of genetic code than most other animals. Isn't that interesting? Because we went through an extinction level event that almost wiped us out at some point in the past.
Yes, that is very interesting. And we did or have 11 species of humans that aren't around anymore, or that are extinct, that we never talk about.
Yeah. Yep.
Like we forget they're even there, but they're gone.
Yep.
It's not unheard of, but I really think that would be something that they're going to do, is they're going to make it so that we can write memory to our genes at the very least, and if not access old ones.
Yep. There's one little example. We're primarily descendants of which? Neanderthals or Cro-Magnons now? I'm just asking because I don't know.
I'm my own super human species.
Well, George Church, of all people, was talking about how there's only limited expression of Neanderthal traits in most people today, but that every once in a while you see a person who's got some of that, some of what were the present genotype of Neanderthals. And they know what they are because they've found bones and preserved genetic material, preserved DNA from Neanderthals in some fossils. Usually it's the ones that are frozen in ice because they're the ones that are preserved best, in case anyone's interested. Okay. And he was postulating or fantasizing, I'm not sure which, about the idea of recovering more of that Neanderthal code. And he speculated about whether there's anybody around today living who's got a significant amount of that Neanderthal code in them alive today. And I think where he was going with it is he was fascinated at the idea of recovering it and introducing it back into the human race for whatever reason. I don't know why, but it was, I've got the link somewhere, but it was a 2-hour talk that they were doing. We don't have time to cover it right now. But anyway, we're in a situation where I don't know how we can keep control of the technology, and things are going to be happening that will produce unexpected results. I think that's a certainty, and that plays against other aspects of the human consciousness, which are the ones that transcend, some of these very physical manifestations. They're more in the range of our mental capacity and our spiritual capacity and our other senses. And none of these guys really are focusing on that too much. It's very much on physical traits and things that are hard-coded or can be introduced into modified hard code in our either our RNA or our DNA. So they're working at that level, and I think that one thing that's a benefit to us is we most of us just know it intuitively. We have transcendent capacities, and that might be where our long-term survivability and our salvation lies. It's just a thought. We're at the end of our 2 hours here for the show. So I want to turn it over to you guys for closing comments. I guess we could wrap up this little article real quick. There's only 3 paragraphs left. TMC, do you want to finish this thing off, and then you can close us out for the
sure
episode? Okay.
Yeah, so interest in the technology among the U.S. Army bureaus has shot up since a secret report by the elite Jason group of military scientists last year received considerable attention among various agencies of the U.S. government, according to an email by Gerald Joyce, who co-chaired the Jason study group in June. A second Jason report was commissioned in 2017 assessing potential threats this technology might pose in the hands of an adversary, technical obstacles that must be overcome to develop gene drive technology and employ it in the wild, Joyce wrote. The paper would not be publicly disclosed, but widely circulated within the U.S. intelligence and broader national security community, the email said.
Yeah, interesting. That's real food for thought.
Yeah.
Yeah. And this isn't this is a 3-year-old article, but this is ever so relevant today. Absolutely. It's more relevant today than when it was authored, to point of fact. Right?
Yeah, I think when you look at the root ideas or concepts that they're talking about here, yeah, absolutely.
Yep. I'm going to throw this link out there in the chat for everybody, and why don't you gentlemen give us kind of a closing remark, and I'll just play the outro. And we want you to be intrigued. We want you to be empowered with knowledge about this stuff, and we wouldn't do it if we didn't. We know how important this is. We do. All right, and I think that if you think about it for a second, you're going to realize the same thing. So, anyway, put that link out there for everybody right now. Okay, guys.
All right, man. Well, it's 2 in a row for you, Indy. So that's good. I greatly appreciate you, man. I think the videos you picked tonight were very good, and that's it. I hope everybody has a good weekend.